Good morning, everyone. So this is the occasion where we are going to hear the lecture by Dr. M.H. Faisal, Professor, College of Agriculture, Bellayani, Kerala Agriculture University, who has kind enough to come over here for the public defense of my student, Dr. Ambati Vasudha, who has excellently done her presentation also. Yes, all the way came from Bellayani for this purpose. So as per university policy, we have requested him to offer one guest lecture. So this has become a normal thing, routine thing in the university that we have to hear whatever may be the discipline, the external examiner or any VIP coming here to deliver a lecture to share their experiences irrespective of the discipline. So you are very fortunate, dear PG students, that it will give some pointer for your carrying out the research experiences or for your future, whatever may be. The clue offered by the external or the subject matter specialist will form a guidance for your career, future opportunities. So in that way, we are glad to receive Dr. Faisal. We welcome you, sir, for your kind-hearted address also. So on behalf of the department, I also welcome our head of the department, new head of the department, Dr. Nelson, who has given the permission, guidance, and everything for this entire proceedings. So as you know very well, that Nelson sir is specializing on biocontrol aspects. Even Dr. Faisal ji is also a specialist of biocontrol, especially on microbial control. His contributions I will tell later. And I also welcome Senior Professor M.R. Srinivasan, honeybee specialist. Now he is a press specialist. And then Dr. Nelson Johnson, who is a PG coordinator. So all the dearmost PG students, welcome you for this formal program. So Dr. Faisal has completed his UG, PG from KAU and PhD from IRI. Of course, he is junior to M.R. Srinivasan sir. So for your kind presence also, we are very happy. So Faisal has specialized on insect pathology, so microbe-mediated plant defense against insects. So his special note of achievements is based on the development of formulations of entomopathogenic fungus, Fusarium pallidorosium against cowpea aphid, and he has characterized the EGT gene in Spodoptera luteura NPV, worked on Erythrina gall wasp and its fungal pathogen association, developed and tested cashew nut shell liquid based botanical pesticide formulations. Of course, the cashew shell, you know that widely used as a medicinal purpose, even for brewing of liquor and all from cashew nut. His work on cashew nut is something different, that he has utilized the cashew nut shell for the botanical as a pesticide formulation for the pest. So currently he is focusing on endophytic microbe-mediated enhancement of plant defense. He has around 25 publications. But 25 means don't look into the number. It is a meritorious publications. So that is his major achievements. So besides that, he has got so many things which not worthy to tell here. But anyway, these are all the highlights of his contributions. So any of the students willing to work or willing to interact in this line, probably you could able to get his number and email for your future collaborations, if at all anything comes into it. So we are happy, sir, to receive everyone here, and we are looking forward to hear your nice lecture, which will become the clue for your PG students especially. Thanks. Thank you, one and all.
To all of you, Honorable Director of this program, Dr. Johnson, respected Head of the Department of Entomology, Dr. Nelson, Professor of this department, Dr. Umapati, who has invited me for the official conduct of the Viva OC plus this interaction. And my dear friend, Dr. M.R. Srinivasan, who was senior to me at my lab at IARI and who was a guiding force for me for the PhD work which I have undertaken on NPV, and students from the Department of Entomology as well as from the other sections. First of all, I thank the Tamil Nadu Agriculture University and Department of Entomology, TNAU, for giving me this opportunity to interact with you. So I am planning to have a presentation on microbe mediated enhancement of plant defense. So from this slide, you can see that Aristotle has said that there is something marvelous in nature. So in nature, we have the clue for everything, including the pest management scenario where we are facing a lot of difficulties. We have several clues to be adopted from nature itself. But only thing is that we should have an open mind and also scientifically approach that so that we will be able to get information from nature itself. So I'll be talking, I have arranged this presentation in two major aspects. So plants itself have the capacity to resist insects. And some of the defense molecules that are produced by the plant directly affects the insects and thereby bringing about its control. And that will form the first part of my presentation, where how can we utilize those plant defense molecules. I'll be concentrating mainly on the biochemical molecules imparting plant resistance, not on the structural aspects. So how these molecules can be utilized for pest management with my own practical experience on cashew nut shell liquid. I will emphasize on the first part of my presentation. And the second part, the same defense against the insects can be triggered even with microorganisms that inhabit various places in the plant. It may be existing as phylloplane microorganisms or rhizosphere associated microorganisms or endophytes. How we will be able to utilize them. These are the two aspects. So plant defense is a multi-trophic interaction which involves both above ground and below ground players. Some of them may be beneficial to the plants, some of them may be harmful. Both these categories of organisms, whether it is microorganisms or macroorganisms, are going to determine or going to play a vital role in the plant defense against insects. So in fact, the three important players here are on one side we have the plants, we have the insects and microbes. They are interacting in diverse ways between themselves. And this triangle of love and hate is what is going to determine the outcome of their association. The plants, as you see here, they are capable of producing defense against insects by volatile organic molecules as well as the plant growth promoting substances. And the insect which attack the plant will be trying to overcome this defense mechanism and try to attack the plant. So that is the beneficial and harmful association. Beneficial association, insects and plants, as you know, there will be pollinators, then insects which are having beneficial effect on the plants. Similarly, microbes will also have beneficial effect on the plant as well as like the PGPRs, but also many of the microbes are harmful, and the harmful microorganisms are tackled by the defense mechanism. And if the pathogen overcomes, then the plant death occurs. And if the defense overcome, the plant will be able to overcome the effect of the pathogen. So defense in plants may be either constitutive, that is always present. The molecule that is offering the resistance will be always present in the plant, irrespective of the insect attack, or many times it is induced to save the energy. That means they will be producing the defense molecule only when it is attacked by the insects. What are the pathways that lead to such a triggered mechanism, and how we will be able to utilize them? Utilize this particular mechanism with the help of microbes that I will be emphasizing later on. So constitutively produced molecules can be isolated from the plant, or their structural mimics can be made, and they can be formulated into a botanical insecticide. There are several such examples. On one side, we will examine that, and the second side, the microbe-mediated induction. So if you look at the global pesticide market, the share of the biopesticides, including botanicals, is very limited. It is only hardly comes to 3% of the total total pesticide market. But if you look at the molecules, there are a lot of possibilities. Around 2 lakh molecules are produced by plants when it is challenged by various abiotic as well as biotic factors, some of which are of use in control. And the secondary metabolites are particularly useful here. Any metabolite that is produced in the plant which does not have a direct bearing on its life. For example, the TCA, the glycolysis, TCA cycle, electron transport, etc., are essential for the plant life. But some of the non-essential pathways are also evolutionarily acquired by the plants, producing certain secondary metabolites which have a very predominant role to play in resistance against pests. So these secondary byproducts or intermediate metabolism will protect the primary metabolism in deterring herbivores and also aid in certain beneficial association with the plants. So if you look at the distribution of molecules, we can see that the classes are terpenoids, alkaloids, and phenolics, which are the predominant categories of molecules that are offering resistance against insects. Of course, a limited amount of steroids, glucose enolates, and cyanogenic glycosides are also there. And the two categories of later molecules, that is the glucose enolates and cyanogenic glycosides, are in fact toxic to the plant itself. But since they are compartmentalized, they will not be causing the harmful effect on the plant. But when it is attacked by a herbivore, that compartmentalization is broken, and that will trigger the defense reaction. So, for example, the glucosinolase, which is a product which is found in mustard family and cabbage, broccoli, radish, etc. So, this will be producing the sorry the isothiocyanates, which when it is acted upon by the enzyme, and the enzyme and the compound which is producing are compartmentalized, making the plant not vulnerable to not vulnerable to the thiocyanate. But at the same time, when it is attacked by the pest, that molecule will be released, and that will be triggering the mechanism. Similarly, the cyanogenic glycosides, which are seen in our cassava plant, is also a potential molecule that can lead to the production of hydrogen cyanide. Here again, the enzyme and is compartmentalized, so it is not toxic to the plant. But when the herbivore is feeding on that, the molecule and the enzyme come in contact, and ultimately leading to the cyanide that is going to cause death of the animals. So the primary metabolism, if you look at there are some important pathways that are leading to specific molecules that offer resistance against insects. For example, the pyruvate and acetyl coenzyme A are important molecules which may enter into the methyl erythritol phosphate pathway or the mevalonic acid pathway to give rise to terpenes. And through malonic acid pathway, it can lead to the production of phenolic compounds. And the nitrogen containing secondary metabolites are also very important alkaloids that are also produced by the shikimic acid pathway, is also producing the phenolic compounds, and the aromatic amino acids plus aliphatic amino acids are going to produce the nitrogen containing molecules. So how these molecules are being utilized to create the defense get modified into defense molecules, that we will examine, and then we will go into how we will be able to utilize them as pesticide molecules. So we will begin with the most important molecule, that is the terpenoids, which occupy a vast share of the molecule that is offering resistance, and that the building block of this is a 5-carbon compound which is known as the isoprene. And through the mevalonic acid pathway as well as the methyl erythritol phosphate pathway, what is formed is two pi-carbon compounds, two isoprene compounds. One is isopentenyl diphosphate and dimethyl allyl phosphate. And when these are interacting, they will be producing the various types of terpenes. The monoterpenes, which have 10 carbons, by combining both these things together, they will produce the monoterpenes. And this again, when it is another isoprene unit is added, then it will form into sesquiterpene. And if two geraniol pyrophosphate are mixed together, that will form the the 30 carbon triterpenoid squalene. And similarly, the again one 5 is added, the geraniol pyrophosphate will be produced. When two geraniol pyrophosphate, it is tetraterpenoids. So depending upon the number of carbon atoms, we have 10 carbon monoterpenes, 20 carbon diterpenes, 30 carbon triterpenes, 40 carbon tetraterpenes, and further additions will give rise to polyterpenes. So all these molecules are produced by different types of plants, that is imparting various kinds of resistance to the plant. For example, if you look at the terpenoids, we can see that there are many terpenoids, like the monoterpene pyrethrum has already been commercialized as botanical pesticide. Then tetraterpenes, azadirachtin has become a very important thing, and the latex essential oils, etc., offer pest control when it is acting in the plants. In addition, the terpenoids being volatile, they will be also acting on the attracting the natural enemies of the pest. When the insect is feeding, the terpenoid will be synthesized, and volatile molecules will be released into the environment, attracting the parasitoids. Phenolics, if you look at it, is a ring structure which cannot be synthesized normally in animals. And the ring structure, benzene ring with an OH group, is the phenol group. Phenol group. And the shikimic acid pathway is the pathway that is producing these phenolic molecules in the plant. And in all the animals, including insects, have to acquire the aromatic amino acids from the plants to synthesize various molecules. And phenylalanine is an phenyl propanoid, that is a ring structure with 3 carbon. And the ring structure with the 3 carbon and 1 carbon are acting as simple phenolics, and their polymers are acting as either lignin or flavonoids. Flavonoids and are important defense phenol molecules that are present in the system. And one more thing which is of importance is that whether it is benzoic acid derived from the phenylalanine, or isochorismate, which is also derived from the shikimic acid pathway, that ultimately can get converted into a secondary messenger molecule, salicylic acid, which has much significance in the triggering as a secondary metabolite messenger, a secondary messenger in the plant system, which we will later look at. So among the phenolic compounds, there are several molecules that has been characterized and utilized as botanical pesticides. For example, you can see the pungam oil. Then we have the rotenone, which is an isoflavonoid, which is again a phenol molecule, and that is also exploited widely as botanical pesticide. So I'll be concentrating I would like to share my experience on a phenolic compound and its potential exploitation that can be achieved by formulating it in a proper fashion. That is the cashew nutshell liquid as a pesticide of plant origin. We have been successful in developing formulation, different kinds of formulation from cashew nutshell liquid, which are used as pesticides. So the pericarp of the cashew nut is filled with the defense molecules in the plant, and that is primarily anacardic acid. And the anacardic acid and its derivatives will be stored in the nutshell as well as the entire plant in lesser quantity, but to a greater extent in the nutshell, offering resistance against various pests. And it is obtained when you process the cashew. This cashew nutshell is a waste. Shell is a waste, and from the shell, during the processing or after processing, we will be able to extract the nutshell liquid. And if the drum roasting technique is practiced in industry, then this will be naturally exuding out from the shells and available as a byproduct in the industry. That is only a very limited use nowadays, and that is available under a cheap rate. So this molecule we have looked into, and we have seen that this is primarily containing the original molecule. When it is intact in the plant, it is containing the anacardic acid. But during processing of cashew, this is getting converted into two molecules, particularly cardinol and then cardol. So this is the ring structure with the OH group, that is the phenol group. And the subsequent side chain is a terpenoid side chain. It is having a terpenoid side chain. So we have tried first with this cashew nutshell liquid under various concentrations, and we have worked out both the LD50 and LC90 values with various pests. My different students of mine has done this work in part, and they have LC90, which can be a field dose that can be taken to the field, varied from 0.25 as low as 0.25 against Apis caxiora to as high as 7.64 against Planococcus cetri because of its waxy covering. So we have analyzed it against various pests of various crops. And subsequently, because it is proved to be a very potent pesticide, we have tried to formulate it in a emulsifiable concentrate formulation. What we have done is that we have determined the HLB value of the uh, hydrophile lipophile balance value of the compound. And based on that, we have selected the appropriate solvent and emulsifiers by trying with the different emulsifiers and blenders, and we could finally come out with a formulation that is stable and also an emulsion that is formed by blooming. Once you put the formulation inside water, that just like a conventional insecticide, it is producing the bloom and giving rise to the uh, pesticide solution that can be applied under the field conditions. So these are the process by which it is being made. And we have conducted all the analyzed the various physicochemical characteristics, and this was found to be a suitable potent insecticide. And also we have tried to make combination with other botanical pesticides like pungam oil, neem oil, etc. And all the test parameters, physicochemical stability and formulation stability, this was found to be excellent. And we have also analyzed the content, what is the active ingredient, and anacardic acid was not present because it is getting modified in the process itself, but it contained the other two molecules. One is the cardol and the cardinol, and that is offering the pesticide efficacy. And these are the different formulations we have developed. In fact, three formulations with that. One is a 20% CNSL alone formulation, and a combination of 20% CNSL and pungam oil formulation, and a CNSL neem oil formulation 10:10. And we have tried it against various pests. And for example, in Bindi, we have tried against all this, and wherein the EC3, that is neem oil, cashew nutshell 10:10 gave good effect against this pest. And against cowpea, particularly against sucking pest, a very low concentration of 0.3% was effectively controlling both the aphids as well as the pod bugs and also the mites. And here you can see that the 0.3% of CNSL EC2 as well as EC1. EC1 is a CNSL formulation load, and EC2 is CNSL plus pungam oil. Both were effective in controlling, though slightly less than the chemical control. Against other aphids also, it was proved to be effective under low concentration. So against sucking pests, comparatively a limited concentration is required, whereas in chewing pests, a comparatively higher concentration has to be employed. Then another potential application of this pesticide was against banana pseudostem weevil, where both as stem injection as leaf axil filling we have tried, and it has found to give satisfactory control of the pest. So from our experience, what we have understood is that the a low concentration of 0.3% CNSL either alone or in combination with fungum oil is affecting in managing the sucking pest, and 1% is effective against both all the formulations as well as EC1 was effective against okra and banana weevils simultaneously, etc. So that is about our experience. So the potential biochemical molecules like the terpenoids, phenolics, and the alkaloids which are present in plants are successful as biocontrol, but uh, can be successfully developed as botanical pesticides. But little effort, if you look into put into that, we will be able to identify more new molecules, and we will be able to come out with excellent botanical pesticides. The opportunity is wide open, and my friends who are young before me, some of you at least can take up this, because that is having industrial application also. We are currently pursuing for a patent for our product, and also subsequently we have to get it registered from the CABRC for wide scale use. But there is also, when it comes to cashew nutshell liquid, when it is highly concentrated material that is available, cardol and cardanol. So the concentration is very critical. If the concentration exceeds above 3 or 4%, then it is likely to cause phytotoxicity. So that is also an important thing that has to be mentioned. So coming to the microbe mediated, how the microbes can be utilized to trigger the production of these molecules in the plant system, so that instead of the molecules being extracted from the plant, we can prime the plant and make it defense ready when the insect is attacking. That is what we are trying in the second aspect. So we have been trying on all these organisms, whether some of the microorganisms are rhizosphere dwelling organisms, some of them living in the leaf foliage, and which is existing as endophytes inside the plant. So in order to understand what it is, first we have to look at the mechanism, because the scientific background is essential when you put up any work. So that has been well established elsewhere. So the resistance is induced resistance, that is, the plant is defense ready when it is appropriately stimulated by the microorganism, and when the pest is going to attack, it will immediately trigger the appropriate defense response. So this is mainly here we are employing mainly non-pathogenic bacteria to either initiate systemically acquired resistance or induce systemic resistance in plant, so that they will become resistant to the pest attack. So these two defense pathways are operated by two different messenger molecules. Uh, and the messenger molecule, one is the salicylic acid is the messenger molecule in case of defense against sucking pest particularly, and also against plant pathogens. As we have already seen, the salicylic acid can be produced from the benzoic acid, or from the phenyl alanine, which is passing through the shikimic acid, and that pathway can produce the salicylic acid, which is acting as the messenger molecule against systemic acquired resistance. On the other hand, the induced systemic resistance is effective against chewing pests particularly, and that is mediated by jasmonic acid particularly, and to some extent by ethylene. So these pathways are operating in different ways in the biological system. And here you can see that the systemic acquired resistance, that is activating the salicylic acid dependent uh, production of uh, pathogenesis related genes against pathogens. So the sucking pest, in fact, is mimicking the activity of a pathogen when it is causing an injury. It is a minute injury which is created by this stylet onto the plant cell, and that mimics the entry point of a bacterial infection or a fungal infection, and the appropriate defense is triggered by the salicylic acid dependent genes. So here, what is isocorosmate synthate and phenylammonia lyase are the two enzymes that are responsible for the synthesis of the salicylic acid, as we have seen earlier. And this salicylic acid will recruit and another protein which is known as the NPR1. And both of them together, when it is binding with the transcriptional factor, that is getting activated, and that will switch on the salicylic acid responsive genes. And when the salicylic acid responsive genes are switched on, they will be able to produce the defense compound. It may be phenolics, terpenoids, or to some extent various pathogenesis related protein. These enzymes which are responsible for the production of the those molecules will be coded by these genes, and they will be under a repressed state. And that, sorry, not repressed, they will be get activated when the salicylic acid NPR is binding. Normally, when the pest attack occurs, that this process will be initiated and the defense will be triggered. Instead of waiting for the pest attack, we will be able to prime the plant by inoculating it with appropriate microorganisms, so that it is in an alert state. And as soon as the pest attacks, the defense molecule will be produced and the insect will be tackled. And plants are able to talk with each other also, whether it is salicylic acid or the uh, jasmonic acid. They are volatile derivatives. By combining with a methyl molecule, methylene can be produced, and that will send volatile signals out of the plant. And the other plants nearby can also sense and trigger the defense pathway can be triggered in them also. So the plants are also able to talk with each other like we are talking, at least with respect to the defense against pests. Then the jasmonic acid is also the molecule, secondary messenger molecule, which is acting against chewing pests. And here the cystamin is a molecule that is produced by the pest attack, and that will bind to the specific receptors, release the membrane lipids, and that will be getting converted into jasmonic acid. And jasmonic acid is the secondary messenger that will switch on the gene to produce the proteinase inhibitors or the appropriate biomolecules that is offering resistance against the pest. So this pathway is normally triggered by the attack of the insect, wounding, plus the signal from the saliva that will activate the octodecanoic pathway, ultimately leading to the synthesis of the jasmonic acid. Unlike the salicylic acid, here normally the defense gene is in a switch off position, and that switching on is acted upon by the jasmonic acid and its recruit. That we will see how it is operating. So when the insect is feeding, a chewing insect is feeding on the plant, that will send the signal, and that signal will produce the OPDA, oxyphytodienic acid, and that will ultimately get converted into jasmonic acid, which is acting as the secondary molecule. And this jasmonic acid will combine with an isoleucine molecule, and this will recruit another protein. Before that, the gene, which is jasmonic acid responsive genes, which are coding for enzymes, or that may be coding for enzymes, or the proteins like protease inhibitors, or enzymes for the secondary metabolite production like terpenes, phenolics, and alkaloids. They will be in a repressed state because it will be bound by another molecule which is known as the JAS. And this JAS removal is required to make the protein for the synthesis of the protein. So jasmonic acid is acting as a messenger molecule that will recruit the SCF protein to degrade the JAS protein. So when this molecule is recruited, the JAS will be removed, and the responsive genes will be activated, and the defense molecule will be produced. And also the jasmonic acid, the volatile derivative is also there, that will be transmitting the signal to the nearby plants as well. So various defenses like the chitinase, peroxidase, polyphenol oxidase, all these molecules can be triggered by the jasmonic acid mediated pathway. So our aim was, so this is the pathway that is operating, either the salicylic acid triggered plant defense production or the jasmonic acid triggered plant defense production. Our aim is to trigger this mechanism not by the presence of the insect, but with various microorganisms that are associated with the plants. So some of the microorganisms are the beneficial. They may be either commensalistic bacteria or mutualistic bacteria without any harm to the plant. Such such organisms are mainly exploited for this purpose. So we have worked on three categories. So we have been trying to with the rhizosphere bacteria as well as rhizosphere bacteria occur in the root zone of the plants, then phylloplane bacteria that occur on the phylloplane, and as well as endophytic bacteria. So rhizosphere, they are occupied by many microbes, of which bacteria predominate, and they are surviving on the root exudates and as well as slough of materials from the roots. And since many of them are known to produce trigger the plant defense response against crop pests, and there are also many products available elsewhere wherein the bacteria are commercialized as biocontrol as products for utilization. So we have tried PGPR with against sucking pests of cowpea again, and then we have seen that the priming with Bacillus subtilis, particularly by seed treatment, seed priming with Bacillus subtilis is offering reducing the mean number of aphids infesting the plant. Also against Streptococcus pedestis, the pot sucking bug also, the another bacteria Serratia marcescens was triggering this response. And Bacillus subtilis was found to be a better candidate for triggering against aphids. And phylloplane bacteria, on the contrary, are stably colonizing the phylloplane of plants, and they will be occupying the space in the trichomes, stomata, epidermal wall junction, etc. And they will remain in the plant stable. Once inoculated, they will be stable, and they will be secreting the chitin and chitinase. So their extraction is also easy because we will provide minimal media into which the leaf can be depressed, and we will provide colloidal chitin as the carbon source. Then that bacteria can be easily isolated. So we have tried with against Epilacna beetle. We have primarily used Epilacna beetle as the test insect, and we have isolated both entomopathogens from the insects as well as bacteria from the phylloplane and tried it against for its pest control. And we have identified that we have cultured many bacteria. We have obtained different isolates of different bacteria, and of which we have evaluated it against the pest attack, and we have found that they are also producing chitin by chitinase by capable of degrading the chitin. And when we have compared its efficacy, two isolates, that is the Serratia marcescens, isolate 3, that is Serratia marcescens, as well as the Pseudomonas fluorescens, which are dwelling the phylloplane area, are successful in reducing all the population of the aphids, the leaf area damage, as well as it is causing mortality on the aphids. Similarly, we have isolated from bitter gourd also. We could get 13 bacteria, and Serratia marcescens was common, and they were found to initiate diseases itself in the Enocephalogna septima as well as Diaphenia indica. Various isolates were producing disease symptoms, and we have also identified them. And one was Klebsiella, and another is Serratia marcescens. And when it was evaluated, it was proved to be colonizing the plant, and also the Serratia marcescens, Klebsiella, as well as Pseudomonas fluorescens. This is Serratia marcescens from the larvae, and this is from the phylloplane of Mammodica charantia, that is bitter gourd. Both are giving better effect, and also Klebsiella was also giving some effect. And this is the over the control. If you see, all these isolates are bringing about pest control effect on when the plant is treated, when the phylloplane is treated with the microorganism. Now we are focusing on the endophytic microorganism, which have much potential, because once it is colonizing, it will stably colonize inside the plant and systemically transported, unlike the other phylloplane or the rhizosphere bacteria. And they are colonizing the plant roots towards inside intracellular space they will occupy, and they will be systemically transported inside the plant, making it effective against pests. So we have developed a protocol for the isolation of the endophytes from the cowpea by heat treatment and then subsequently extracting to get the endospore forming endophytic bacteria from these compounds, and we could successfully isolate the many isolates from vegetable cowpea, and altogether we have already obtained 49 isolates, and these 49 isolates were brought to pure culture, and testing was a difficult thing. So here there were some practical difficulties. When we tried the leaf dip method, the effect was not pronounced. And also when we attempted detached leaf assay, that was also giving not distinct effect between the isolates. This is because here in both the cases we are detaching from the plant, and the plant is not live, and the secondary metabolite production is not happening in an appropriate level. So we have tried in plant analysis. That is, the plant will be inoculated with the bacteria, and it will be live plants will be kept, and we will be releasing the insect, and we will be analyzing the situation. And here we will be scoring the damage intensity, and when we have analyzed the damage intensity score, we could obtain distinction. And you can see some of the isolates are very superior in reducing the disease index. And we have tested 49 isolates in 7 batches, and we could get promising isolates by this trial. And then against chewing pests, we have conducted an in-plant analysis. A cage type of thing was made, and the larvae were released inside the cage, inside in the live plant treated with the bacteria, and then we have analyzed the effect antioxidant action as well as the relative growth rate. And there we have found that these bacteria, 2 isolates, CPB1A and CPBA, are reducing the relative growth rate of the insect to a significant level. And also this is reducing the feeding activity of the insects as well as and this we have also analyzed biochemically, and it is directly linked with the phenol content. And you can see that the phenol content in these 2 isolates are getting increased, and total phenol content in these treatments, CPV1A and CPV8 and the Bacillus subtilis was lower than the other, was higher. The phenol content was higher when either in the before release, after release, and after 48 hours after release. That indicates that the triggered mechanism is happening with the bacterial inoculation, and once the first is inoculated, the secondary metabolite production is enhanced to a greater level, bringing about resistance against the pest. So in conclusion, there is a possibility of exploiting the compounds directly or with the help of the microbe mediated way. The plant defense molecules can be enhanced for eco-friendly pest management. And in future, we will be able to identify large number of such strains, and also we will be able to go for genetic modification of strains if required. Such a work has already been undertaken elsewhere. This is not our work. This is been proved. So in this line also people can think of and work out, and so that they can bring about successful control. So this is what in nutshell I wanted to share with you all from our experience on these 2 aspects. Thank you very much.
This cashew nut cell liquid, what is the cost of that formulation, sir?
The cost, apart from the cashew nutshell, which is procured from the industry, which may be costing around 60 rupees per liter, there is not much cost. The other things are the solvent and also emulsifiers, which are comparatively cheap and also used in a very limited extent. The percentage is also less. In fact, altogether it will be for 1 liter. Once we standardize the procedure and everything, it is hardly 100 rupees per liter. Pardon?
Thermal toxicity.
Dermal toxicity. Okay.
Skin, it cause blisters.
Okay.
We have seen that in
yes, phenolic. That is a very important, pertinent question. The cashew nutshell liquid is containing high phenol, that is cardol or cardanol. It is present in high amount, and that is in fact the phenols are toxic molecules. But in the formulation, we are dissolving the cashew nut shell in a appropriate solvent. Here we have used isopropanol as the solvent. Once it is getting dissolved, that toxic action is getting reduced. And to the application side, we are using only a very, very low concentration of the cashew nut shell liquid. For example, 0.3% or 0.1% against sucking pus. And for chewing pus, up to the maximum of 1%. So at the application, once it is mixed with water, it is forming an emulsion and it is not toxic when it is falling onto a hand. But on the plant, there is some danger. If the concentration exceeds, say 3% or 5%, then some phytotoxicity has been observed. And also, it is interfering with the auxin. Auxin usage in the plants, and it can initiate senescence type of symptoms and shortening of the plant if the concentration exceeds. So that is one of the limitations when we use it as a botanical pesticide. We can supply to farmers, and we can recommend the dose, but sometimes some farmers accidentally or unknowingly, if they increase the concentration beyond the recommended level, not the recommended level, beyond 3%, then there can be a potential danger. So we are also working out on that. We are trying to reduce the amount of cashew nut shell and trying to compensate the effect by providing other botanical oils like neem oil and the pongam oil, etc., to overcome that effect.
Good afternoon, Dr. Faisal. So I'm very happy to see you again.
Thank you.
My college days, after our college days, and again, I think you were here for my students' Viva.
Yes.
Ishantej Viva. So nice. You are doing a wonderful work, and you have shared it in a nutshell about the nutshell liquid. Yeah. Okay. So I have a question. What is the safety of these products to pollinators? Like one is the compound and the nutshell liquid, and again the other microbes that you are using.
Okay. Nutshell liquid, we have tried it with the pollinators. Only honeybee we have tried. All the honeybee species we have given at the recommended and the double recommended concentration, they are found to be safe. The microorganism we are yet, because this is in the initial stage, then only we will be trying for its safety with other. But microorganisms, those microorganisms are innocuous and they will be surviving either in soil, phylloplane, or the as endophytes, not coming in direct contact with the other insects. Only thing is that they are triggering the biomolecule production in the plant when it is challenged by an insect.
Fine. So actually, for the information of the students, so we did our PhD in the 90s. Like I completed in 93, and Dr. Faisal completed must be 95. 97. Yeah. Actually, we worked in a lab which was specializing on NPV at the time, nuclear polyhedrovirus, and also the molecular characterization of this virus. So I worked on gratis NPV, and Dr. Faisal worked on Spodoptera NPV. So actually, he was trying to actually delete a gene. It's called EGT gene. Normally, that is responsible for resistance in late larval stage. So that work he has done. So it used to be a prestigious laboratory, and even molecular biology laboratories were not so equipped in those days. And so students from molecular biology lab also used to come to our lab. So I'm remembering the days when we were
good old days.
Yes. Thank you.
Anybody working with botanical insecticides? Any students?
Sir, I am Mohan Kumar. So can you hear me, sir?
Yes, sir. Yes, sir.
Yes, sir. So thank you very much, Dr. Faisal. It's a wonderful lecture for the benefit of the students. Thank you.
Thank you, sir.
And so you informed about the cashew nut shell, so and its utilization. So in that, so you are saying that you are going to patent that.
Yes, sir.
So already there are many patents in US and others, some 50 or 60 years back. Because of the toxicity and also the dermal toxicity, they have, I think, dispensed. As like in the initial stage of the neem and other thing, even before neem, the shell oil was tried and then it has a potential against nematode as well as for insects, but the formulations are not came to market because of the toxicity, and the many patents are available for that insecticidal activity. And the second one, you have explained about the microbes which are effective against that. So in 2 or 3 microbes you highlighted, one of them is Serratia marsans. So again, here also a potential danger. I think everybody is saying that now Serratia, we have to discontinue the work because of they are affecting the lungs of human beings. So if it is handled, so if it is in a normal, if it is in soil, it is okay. Natural resistance, if it is given by them, it is okay. But when we are going to spray it as a formulation, then again, it is also pose somewhat a biosafety level. So for registration, it will be a problem. I think, what are your views on this?
Okay. Yes, sir. What you have raised are very important and pertinent questions. And the cashew nutshell liquid, of course, in a highly concentrated form, when it is not dissolved in an appropriate solvent, it is causing toxicity. But once it is getting dissolved, what we have observed is that once it is getting dissolved in the solvent, the properties are changing. Not the insecticidal properties, but the other properties. The viscosity itself is becoming very light and it is flowing just like a liquid. And when we formulate it and then the small amount of, we have not tried with extensive dermal toxicity studies. That is, of course, we are at present trying to patent the methodology of production. As a pesticide, of course, there are different other steps which we have to encounter and overcome. But what we have noticed is that this formulation, the formulated product, is not that toxic as in the case of the unformulated product. One thing. And the other second aspect that you have pointed out is also very, very crucial. Serratia marcescens is an opportunistic human pathogen also. In immunocompromised people, that is known to produce certain ill effects. But what we have observed, this is a preliminary work. When we extracted from the natural world itself, many of the bacteria were Serratia marcescens that we obtained, either from the phylloplane or from the diseased insects. It was mainly Serratia. So that we also taken up further investigation, because that is already present naturally. What we are doing is that we are priming the plants. Just at the for endophytes, we will be treating at the seed level only. Subsequently, there is no treatment. Sterilized soil for experimental purpose, into sterilized soil, the primed seeds are put. After that, we are not at all releasing the pathogen, the organism. The organism is going to exist as an endophyte, which otherwise also naturally it is occurring in many plants. So that is what we have been trying. But of course, when it comes to the formulation side, then there will be practical difficulties. There, our candidate microorganism is mainly Pseudomonas fluorescens, which does not have such effects, though it is inferior to Serratia marcescens in our experiments. Sir.
Thank you. Thank you very much.
Good morning, sir. Thank you for your lecture. This is Piridhiraj, third year PhD student, sir. Could you hear me?
Yes, sir. Yes, sir.
Yes, sir.
Yes.
Sir, in one of your slides, you mentioned that after infestation, the phenolic contents are getting increased, the total phenols. So usually, theoretically, we study phenols are a constitutive defense.
Okay.
So in such a case, after infestation, it has to be reduced.
When it is
Can you explain?
the total phenol content, what we have noticed is that it is primed with a microorganism. Priming means we are over the initial steps of the secondary messenger molecule production is already undertaken in the plant system. So without much delay, when the pest is attacking, the phenol content, what we have noticed, but we have not worked out the biochemical pathway how the phenol is getting increased. From our we have analyzed several biomolecules, but what we have observed is that phenol content is getting increased in the when it is not inoculated with the not challenged with the insect, as well as when it is unchallenged in the plants which are treated than the untreated. That means what we presume is that the secondary metabolite production is in a primed state so that it can express when the pest is attacking. Normally, the phenol content is required to some extent for the pest, but when it is excessively produced, then it is producing the ill effects. That is what we have observed. That endophyte study is in the initial stage only. We have not even identified those bacteria. We have isolated it and we are testing it. And once the some isolates were proven effective, then we will get it identified and further work on that.
Thank you, sir.
Any students working on botanical pesticides or any molecule, anything like that? Nobody is working on that line. That is a potential area you can think of. At least your juniors can take up some work in microbe-mediated enhancement or the botanical pesticide utilization. Unconventional, not at utilized ones. Okay, then if there is no further queries, thank you very much for patiently listening to me. And I thank all the authorities of Tamil Nadu Agriculture University in giving me an opportunity to interact with you. It was a wonderful experience.
Examiner is Dr. Faisal from College of Agriculture, Velayani. My dear colleagues, my dear students, good afternoon to each and every one of you. First of all, thank our external examiner who has accepted our invitation and made a presentation today in an elaborate manner on botanical insecticide and the microbes involved in the plant defense system. I personally thank on behalf of our administration also, I thank Dr. Faisal for his excellent presentation as well as for conducting the Viva in a nice way. Thank you very much, sir. And I thank Dean SPGS, our Director CPPS, for encouraging us to conduct these type of seminars when our external examiners are coming from other parts of our country. So now you had a chance to meet people from different parts of the country because of the encouragement from the administration side. Dr. Vice Chancellor and Dean SPGS, they are all encouraging these types of seminars. Many online seminars are conducted by Dean SPGS. Kindly make use of that seminars. Then I thank our professors, Dr. Emma Srinivasan and other professors who are online for their presence on this day. And I thank the PG coordinator who has arranged these type of final iOS examination and involved in all students' activities. I thank PG coordinator. I thank Dr. Umabadi, who is the guide of today's Tambati, and the Tambati, the student from Andhra Pradesh. She has actually presented very nicely. Dr. Umabadi and his team members, advisory committee members, they actually guided that student to produce a very good thesis, and she has presented very nicely today. And I thank Dr. Umabadi and advisory committee members for encouraging the students to do a very good research work on that aspect. Finally, I thank all the students for your presence in this seminar. Thank you, one and all. Thank you very much.